Hello, my name is Jan Michael Peters. I'm a senior scientist and scientific director at the IMP, the Research Institute of Molecular Pathology in Vienna, Austria. And I would like to talk to you about the role that the cohesin complex and the DNA binding protein CTCF have in folding DNA in mammalian genomes. Now, in this second part of this lecture, which has two parts, I would like to discuss with you what the evidence is that we have in the field that cohesin has such a DNA folding function. Then I would like to discuss with you how we think the CTCF protein contributes to this process. And I would like to discuss with you the loop extrusion hypothesis, which provides a very interesting novel view on how DNA loops might be formed. Now, in case you have seen part one of this lecture series, you might remember that one can see loops relatively easily by light microscopy in very specialized meiotic chromosomes, the so-called Lamprecht chromosomes. But it turns out it's much harder to visualize loops in somatic interface nuclei. Best one can do this for one or two or a few at a time by using hybridization of fluorescent labeling techniques. But it turns out that one can identify such loops, long-range interactions, by a different technique called Hi-C, which was developed around 2009 by Aris Lieberman-Eiden, which uses a formaldehyde cross-linking in cell nuclei to physically connect sequences that at the point of cross-linking had been in close proximity. And by processing this cross-linked DNA through multiple steps, one can then use next-generation sequencing to identify which sequences had been cross-linked, i.e. had been in close proximity. And these long-range interactions, one can then visualize in heat maps, as one is shown here, where typically one shows DNA sequence on the x-axis and the same sequence on the y-axis, so that one can visualize by intensity of color which regions in this particular locus interacted with other regions in the same locus. And one can do this for the whole genome in one Hi-C map, or for a single chromosome, or one can zoom into a particular locus as we did here. Now, if one looks at very long-range interactions, which are not shown here, one can visualize the compartments that are briefly mentioned in part one of my lecture. But these are not thought to depend on cohesin, so I will not talk about them today. But if one zooms in further, as we did here, one can see two different types of patterns. One of these appear as pyramid-like structures, so-called topologically associating domains, within which many sequences seem to be able to interact with other sequences in this particular domain, first described in 2012 by Bing Ren and Edith Hurt. And a few years later, by increasing the resolution of this technique further, Aris Lieberman-Eiden could show that in addition, one can see also discrete dots in these Hi-C maps, which we refer to as loops. It's important to note that these loops might, in mechanistic terms, not be very different from the interactions that constitute TADs. But these are the two types of patterns that one can identify, which do represent folded DNA within interface chromatin. Now, the question was, do these interactions depend on cohesin, as we had hypothesized, and I explained in part one of this lecture series. And to be able to test it, it was important to inactivate cohesin very efficiently. Otherwise, one wouldn't see whether these structures really depend on these proteins or not. And we and others did this by using a technique called auxin mediated degradation, where one is fusing a sequence domain to a protein of interest, in our case, a subunit of cohesin, that can be recognized by an adapter protein that, under the right conditions, can then target the protein of interest to a ubiquitin ligase, which can then multi-ubiquitinate this protein and thereby target it for destruction by the 26S proteasome. And this particular tagging system can be controlled by the absence or presence of the plant hormone auxin, which is needed for activation of this particular ubiquitin ligase system, which is being used here. Now, we did this for the cohesin subunit SCC1, which we tagged with such an auxin-inducible degron tag, AID, but in addition also with GFP, so we could visualize it by fluorescence microscopy in cell culture. And when we added now auxin to such cells which contained only SCC1 fused to GFP and ARD, achieved by CRISPR genome editing, but no other untagged alleles of SCC1, then we saw that the GFP signal disappeared very rapidly, quantified here. And this disappearance of GFP was indeed caused by degradation, as could be seen by Western blotting, where the SCC1 protein disappeared both from the chromatin pellet, but also from the supernatant fraction, whereas other subunits of cohesin remain stable, for example, SCC1, but interestingly disappeared, were released from chromatin, as you can see here, at the same time as SCC1 was degraded, exactly as one would have predicted, since we know that these are subunits of the same cohesin complex. So this turned out to be a very efficient and also rapid, by the way of inactivating cohesin. And so the question then was, what would now happen to chromatin structure? What will we see in high C experiments with respect to the presence of TUDs and loops? And the phenotype is really dramatic. As is shown in this slide, on the right-hand side, you can see the same chromosomal locus that's shown on the left, but now 3 hours after addition of auxin to the cells that only have cohesin, which is degradable. And you can appreciate that the majority of interactions that constitute TUDs and loops has gone. They disappeared, implying that cohesin is indeed needed for these long-range interactions. And we've also looked at earlier time points by high C and can see, consistent with the disappearance of SCC1, that these structures begin to diminish already after 15 to 20 minutes. So clearly they depend on cohesin. That's one reason for believing that cohesin is important for folding DNA in mammalian genomes. The other reason for believing that comes from a very different type of experiment, which initially we did for a different reason, where we wanted to know why cells have an elaborate system by which they can remove cohesin from chromatin again using the release factor WAPL, which I introduced in the first part of this lecture, which we think is a protein that binds to cohesin and opens the ring structure, thereby enables it to dissociate from DNA again, which clearly happens to complexes that have not established cohesion in G1 phase, for example. Now, because WAPL is a release factor, we had expected that its removal would now stabilize cohesin on chromatin. It would have a longer residence time. And we also expected that as a result of that, there would over time be more cohesin accumulating on chromatin, because there's always an equilibrium between soluble and DNA-bound cohesin. And if we removed WAPL, then of course the prediction was it would continue to be loaded onto DNA cohesin, but would no longer be released. So it would have a long residence time, and it would be there in higher amounts. And that is indeed exactly what we found. But what we had not expected was that these changes would have dramatic effects on chromatin structure and also on the localization or the distribution of cohesin within the nucleus. And that's shown on this slide, where on the top you can see a mammalian cell nucleus stained with DAPI, so the DNA is stained. You can see a relatively amorphous staining pattern, which is typical for a cell in G1 phase, which this is. But after inactivation of WAPL, now we can see that the DNA had begun to become condensed, as if it had entered prophase, early mitosis, which this cell clearly hadn't by all sorts of criteria that we looked at. Even more surprisingly, we found that the distribution of cohesin was dramatically changed. Normally, as seen by fluorescence microscopy, cohesin would be distributed on chromatin relatively evenly, as you see here in this control cell. But after WAPL depletion, now it had changed localization and accumulated in axial domains of chromatin territories, which from hybridization experiments we believe are running from one telomere, so from one end of the chromosome, if you wish, to the other. And we called these axial staining patterns vermicelli, which is Italian for little worms, because the student in my lab who discovered them was Italian, Antonio Tedeschi. So that's what we call these structures. Now, That was really interesting. It reminded us of how complexes that are related to cohesion are distributed in mitotic and meiotic chromosomes. In mitotic chromosomes, the condensing complexes are also adopting an axial distribution. And in meiotic chromosomes, there are different types of cohesion complexes, specialized meiotic forms of cohesion that also form similar axial structures. And in both cases, it is believed that these axial structures form the base of chromatid loops. So based on that, and based on our hypothesis that cohesion might be folding DNA into loops, as I explained in part 1 of the lecture, we hypothesized that these vermicelli, these axial domains, might also represent the base of chromatin loops in WAPL-depleted cells. Now that was very exciting, but the real question, of course, was how would the depletion of a release factor, which leads to stabilization of cohesion or chromatin, lead to this dramatic relocalization of cohesion into vermicelli, and how would it then somehow lead to compaction of DNA? That was really unclear. And it was Leonid Miny, a polymer physicist at MIT, realized that both these phenotypes could be explained by a very interesting hypothesis called the loop extrusion hypothesis. This hypothesis has been postulated multiple times over a number of decades independently, starting with Arthur Briggs and others working on enhancer-promoter interactions, who were faced with the question, how could a distant enhancer element in DNA find its target gene promoter? And they speculated, they hypothesized that maybe it would find it by what today we would call an extrusion mechanism. I'll explain in a second what that is. Then Kim Naismith postulated that condensate would form loops by such a mechanism in mitotic chromosomes, and by extension maybe also cohesion would do something similar. And then after that, Leonid Miny and several others, Iris Lieberman-Eiden and John Marco, also came up with this idea. Now the Miny lab realized that this hypothesis could explain our WAPL depletion phenotype because the loop extrusion hypothesis posits that maybe two different sequences would specifically be brought into proximity, not by diffusion, that's hard to imagine, but by a mechanism where a hypothetical extrusion factor would land somewhere on the genome, and then by an unknown mechanism would pump DNA into a loop-like structure until it would reach eventually the anchor sequences that would be defined, or would be the sequences that should form the base of such a loop. And if one imagines cohesion could be such a loop extrusion factor, it could explain why WAPL depletion would give these really unusual phenotypes, unexpected phenotypes, in the sense that if WAPL was present, we would know that it would limit the residence time of cohesion on chromatin. And so if cohesion was forming a loop by extrusion, the lifetime of such a loop, and therefore also its length, would be limited, because every couple of minutes or so, WAPL would release the cohesion complex again, and the loop would fall apart. But if WAPL was now depleted, cohesion could no longer be released. It would have a long residence time, and it could now form very long loops, longer than usually. And in theory, they could be so long that eventually the cohesion complexes could bump into each other, and that could explain why we saw them now all of a sudden accumulating in axial domains within interphase chromatin territories. And at the same time, the formation of long loops could explain why chromatin became compacted, because it is thought that formation of long loops, that's been supported by in silico simulations and polymer modeling, are causally responsible for chromatin compaction. Now, this hypothesis made the very interesting prediction that when we depleted WAPL, we should now be able to detect more long loops, more long-range interactions by Hi-C. And both my lab and also the one of Ben Rowland have tested this and indeed found that that's the case, as is shown in this slide, where on the left you see again a Hi-C matrix from a control cell. But then on the right-hand side you see a Hi-C matrix from a cell from which WAPL was depleted. And I hope you can see that now there are additional long-range interactions in the WAPL-depleted cells that cannot be detected, at least to the same extent, in the control cells, implying that now more long-range interactions had been formed. We showed that these, again, depend on cohesion, of course. Now, in the first part of this lecture series, I explained that initially we had speculated that cohesion would be the molecule that mediates long-range interactions to form chromatin loops, but it would be CTCF, or its ability to recognize a particular consensus sequence in the genome, that would specify the identity of these loop anchors. Now, if cohesion was mediating loop formation by extrusion, the question is what could be the role of CTCF in this process? Now, obviously, CTCF could function in this process as a physical boundary. Everything that we know about these loops could be explained if one assumes that extruding cohesion complexes were stopped in the extrusion process once they encountered CTCF when it's bound to DNA. And that would, in fact, very nicely explain the observation that cohesion accumulates, as one can see by chromatin immunoprecipitation, at CTCF sites, and therefore seems to co-localize, at least if one analyzes large populations of cells. So the loop extrusion hypothesis would predict that CTCF must be a physical boundary for extruding cohesion complexes. Now, what is very interesting in this context is that the binding site or the consensus site for CTCF is not a symmetrical sequence, but asymmetric. It's not a palindrome. And for that reason can exist in two different orientations in the genome, implying that CTCF can bind in two different orientations. Now, that in itself is not unusual for DNA-binding proteins, but what is very unusual is that a number of labs have observed that those CTCF sites that form the anchors of cohesion-mediated loops are always pointing towards each other in their orientation. And that implies that CTCF may not only function as a simple physical barrier, but may function as a boundary in an asymmetric fashion, implying that if cohesion was arriving from one side at such a DNA-bound CTCF molecule, it might block extrusion, but if it's arriving from the other, it might not. And in fact, Maciek Zakzak in my laboratory has recently been able to reconstitute part of this boundary function using recombinant CTCF and cohesion, and has seen that indeed, under in vitro conditions, CTCF is sufficient to block translocating cohesion complexes that arrive at a CTCF site from one side, but cannot block cohesion if it's translocating along DNA from the other side. And how that works mechanistically, what the structure of CTCF looks like to be able to achieve this unusual symmetry will be a very interesting question to study in the future. Now, that in itself then represents another complication, because if these CTCF sites function as boundaries for loop extruding cohesion complexes, why would then the loops become longer if WAPL is depleted? And we don't know the answer to that, but one has to postulate that they do become longer because these boundary elements, these CTCF sites, are only transient boundaries, either because CTCF dissociates and rebinds, and while it does that, cohesion can go past and form a longer loop, or perhaps because cohesion can somehow at some point jump over such a CTCF boundary and thereby form a longer loop. So we think of them a bit like red streetlights in a traffic system where cars would accumulate in front of the red streetlight, but of course at some point the streetlight would switch to green and then the cars would move on. And maybe that's how cohesion somehow analogously behaves with respect to CTCF as a boundary element. But what is interesting is that the longer loops that we see after WAPL depletion, that's shown here, indeed occur as discrete dots on these high C matrices. So it's not a range of long-range interactions, it's very discrete, longer loops that can be seen. And if one looks at the sequences underlying the anchors of these loops, it turns out that each of them represents a CTCF binding site, as if cohesion, now having a long residence time, would be able to sit long enough on DNA to go beyond one CTCF boundary, but then would stop at the next one until it could go over that, and then would stop at the next one. Again, in analogy to the red streetlight model. Now, if this was all correct, then this loop extrusion hypothesis would make a number of predictions, namely that the binding of cohesin that we had initially identified by chromatin immunoprecipitation as being found in very discrete peaks, I showed that an example in part 1, would in fact not represent how cohesin really behaves on genomic DNA, but would basically be an image that emerges from superimposing the behavior of many cohesin complexes in large populations of cells. And that in reality, cohesin would be highly mobile in the genome. It would be extruding loops all the time, and only the complexes that would be stopped by CTCF would then be present in these peaks that one sees by chromatin IP. So the hypothesis was that cohesin would be highly mobile, but would be constrained once it hits a CTCF site. And another important prediction would be that CTCF itself would not be important for the long-range interaction, but would define where they are being formed. It would be what recognizes a particular sequence and thereby specifies what the loop anchor should be. And so both of these predictions have been tested by us and others, of course. First to the question, is cohesin mobile in the genome and is it constrained by CTCF? To test this, we have depleted CTCF, initially by RNAi, in this experiment by using a mouse model provided by Niels Galliard, where inducibly the CTCF gene could be deleted. And when we did that, we indeed found that cohesin no longer would now accumulate to the same extent at CTCF sites. That's shown here, where you're looking at chromatin immunoprecipitation results, and you're looking at one of thousands of CTCF sites that are present in mammalian genomes. And you see that at the same site, cohesin can be detected, also by CHIP, chromatin IP, provided CTCF is there. But if CTCF is depleted, because the gene has been deleted, now much less cohesin accumulates there. And that's true not just for this site, but for thousands of sites. Now, interestingly, what we had expected is that under these conditions, cohesin accumulates at different sites. As you can see here, it often accumulates at the promoter of genes at transcription start sites for reasons that are not well understood. But implying that indeed cohesin is highly mobile in the genome and not statically sitting at one site. Now, that can be even seen more impressively or more clearly in cells where not only CTCF has been depleted, but also the release factor WAPL. And an example for that is shown here, where again you're looking at chromatin immunoprecipitation results, where you're seeing one of many thousand CTCF sites, which normally co-localizes with cohesin, provided that CTCF is there. As I just showed in the previous slide, if CTCF is depleted, then cohesin is no longer accumulating there to the same extent, but now instead accumulates at transcription start sites. One is here, another one is shown here, there. But what is really interesting is what happens if now, in addition to CTCF, this condition, also WAPL is depleted. So that now cohesin has neither its normal boundary nor its short residence time, but instead a long residence time of chromatin. And now you can see a very dramatic effect, namely that cohesin accumulates in very large regions, not looking like a regular peak. And these so-called cohesin islands, as we call them, are typically found at the 3' end of active genes, in particular at the 3' end at loci where two genes converge on each other. One is being transcribed from left to right, and the other from the other strand from right to left. And at these convergent locations, cohesin now is accumulating, provided there was no CTCF as a boundary, and WAPL was depleted, so it has a long residence time. Implying that, yes, it must be highly mobile in the genome. It's probably moving around all the time, but normally is seen accumulating in cell populations at CTCF sites, but then can go to very different places if these boundaries are gone. So that's clearly consistent with the loop extrusion hypothesis. Now, what do we know about the role of CTCF in this process? The loop extrusion hypothesis posits that it would not be needed for forming long-range interactions, but instead would specify which sequences form these interactions. And indeed, the data that we and others have are consistent with this idea. Again, if we look by high C, in the presence of CTCF or in cells depleted of CTCF, this experiment was not done genetically, but again by using auxin-induced degradation of CTCF in this case. First of all, we can see that superficially the two high C patterns look very similar. You see there's still lots of interactions present, and one can analyze this also genome-wide by plotting all the interactions. This is shown in this graph down here on the y-axis as a function of the genomic distance on the x-axis. And this graph must look to you like just one line, but in reality it is two lines superimposed, one representing the distribution of interactions in the control cells that have CTCF, and the other representing the interactions in cells depleted for CTCF. So the result is there is no change. The interactions are still all being formed, their number is roughly similar, and their length distribution in the genome is also roughly similar. So that clearly tells us that CTCF can't be essential for the formation of these interactions. That's because we think cohesin is mediating them. Now, but if you look more carefully, you can appreciate two differences, and they're interesting. First of all, one can see, if one looks very carefully, as explained at the beginning, that at the corner of these TUDs, there are often these dots, which we think are loops that have been formed. And they can no longer be clearly detected after depletion of CTCF, as you can see here. The arrows are pointing at this. And genome-wide, one can quantify this using special algorithms that's shown in the histogram on the bottom, where you can see that the number of loops one can algorithmically detect after CTCF depletion is much reduced. And a similar phenomenon is seen if one looks at the boundaries between TUDs, which in the presence of CTCF appear as relatively distinct and sharp boundaries, but after depletion of CTCF now are much more fuzzy. And that can again be genome-wide analyzed algorithmically as a so-called TUD insulation score, and one can see that this is much reduced after, not abolished, but reduced after CTCF degradation. So what these results imply is that CTCF is indeed needed for the insulation of TUDs, presumably because it's normally sitting there and functions as a boundary for extruding cohesin complexes. And the fact that one can no longer detect so many loops in the absence of CTCF is presumably not because these loops are not being formed. We think they are all there, but no longer they are confined exactly to the CTCF binding site as a sequence anchor, because without CTCF, cohesin can now go a little further than normally and would form loops that are less specified, less defined in these large cell populations that are being analyzed here. So these data are consistent with the idea that cohesin is essential for TUDs and loops. I showed you that at the beginning. It forms clearly longer loops if WAPL is depleted and cohesin now has a longer residence time. And clearly, cohesin must be highly mobile in the genome. And what we typically see in chromatin IP experiments is just the average of where most cohesin has accumulated, but not really showing us where in individual cells cohesin would be present while it's extruding loops. And for CTCF, the data are consistent with the idea that it's not essential for the actual long-range interactions, because these are mediated by cohesin, but is needed to specify them by telling cohesin where to stop the loop extrusion process. And that would then form these less sharp TUD boundaries and would result in the reduction of loops that can be detected. Now, these phenotypes, in the case of CTCF depletion, are clearly not null phenotypes. You can see there are still some TUD boundaries. And so an important question for the future will be, is that because CTCF wasn't fully degraded? We think it is not by this AID system, so that could be one reason. But of course, it could also be that there's other boundaries or other molecules that contribute to a boundary function. And that's why CTCF depletion has a partial loss of TUD insulation and not a full phenotype. That remains to be explored. Now, towards the end, let me briefly discuss with you what we know or don't, about the really important question then how cohesion in mechanistic terms might be forming chromatin loops, and in particular whether it does so by loop extrusion. Now, the short answer is we don't know. But before I'll summarize for you what we know about cohesion, let me turn to cohesion's cousin, the condensin complex, which, as I mentioned, is thought to be responsible for forming long chromatin loops on mitotic chromosomes and thereby condensing these chromosomes. And very recent work from Christian Hering and Kees Dekker has shown that if one prepares or isolates these complexes, the condensin complexes, from budding yeast and uses them in similar assays, 12 microscopy assays to the ones I've just shown, one can indeed see that these yeast condensin preparations have an activity which is able to extrude a loop. That's extremely exciting, was published last year. That's just to remind you that cohesion and condensin are complexes are related, and they both accumulate in axial elements under the right conditions. Now, if we watch this movie, you can see here a DNA molecule that's tethered at both ends to a glass surface under buffer flow, and you will see in a minute that one arm of this synthetic DNA loop, if you wish, is shortened at the expense of a new loop that's being formed here, coming out of this doubly tethered DNA molecule. The idea being that condensin must be sitting here in the center of this doubly tethered DNA molecule and must be pumping DNA from one side into a loop structure. And that's schematically shown in this illustration from the paper from Kees Dekker and Christian Hering, according to which condensin must be sitting on this doubly tethered DNA molecule and must be pumping DNA from it into such a loop-like structure, as you can see here. Now, such an activity has not been seen for cohesion yet, but this result clearly illustrates that members of the SMC family must have activities that are able to form DNA loops. Now, that raises the really interesting question whether cohesion can not only function as a topological linker, as which it is thought to function when it mediates cohesion by embracing the two sister DNA molecules inside its ring structure, but whether it can actively form chromatin loops and perhaps therefore is a molecular motor, more related to other motor proteins like kinesins or myosins or dynein, and whether it could use its ATP binding and hydrolysis activity, which it clearly has, for that purpose. And very recent experiments performed in my laboratory by Maxim Molotov and Benedikt Bauer have indeed provided evidence that cohesion has such a motor function. This evidence comes from experiments in which we have tethered cohesion to a glass surface with one of its subunits, and then connected one of the other SMC subunits to a long handle, a coiled coil, which again is connected with a bead, the position of which can be controlled by a laser beam. And this so-called optical tweezer type of experiment allows, at the piconewton scale, a measurement of forces that are generated by proteins, such as motor proteins. And it turns out from these type of experiments that indeed, under the right conditions, cohesion is able to perform a power stroke, to perform mechanical work, which we think it uses to bend DNA. And from these experiments, which are not published yet, we have reasons to believe, therefore, that cohesion can not only function as a passive linking molecule, which it is what it seems to do during cohesion, but instead is an active motor and uses that activity to bend DNA. And we've also found that the ATPase activity and the NRPBL protein, which has previously shown to load cohesion onto DNA, are needed for this motor function. And so that quite radically changes our view of how cohesion is functioning at the mechanistic level. But at the same time raises many new questions, in particular the ones that I have listed here, namely how cohesion could bend DNA. For that, it would have to be able to bind to it directly, And so we are trying to understand how it can do that. The topological interaction that has been described before would obviously not be sufficient for such a bending activity. We would very much like to understand whether this ability to bend DNA and function as a motor is important for structuring chromatin into loops and cells. Again, that needs to be tested. And obviously it would be very important to know whether this motor activity that we can measure by optical tweezer experiments is required for loop extrusion for the type of DNA loop formation that Kees Dekker and Christian Hering have observed for yeast condensin, but no one has observed for cohesin yet. And so these are very exciting questions for the future that I'm sure the field will answer in the coming years. Now, I would like to thank you again for your attention, but also my former and past members of the lab, which have done fantastic work, which has contributed to the knowledge that I've summarized here, as well as to all other colleagues in the field that have contributed to what I told you about today. And last but not least, to our shareholders and funding agencies which have made our work possible. Thank you very much.